we looked at one type of hidden messages, which was just a gene, a region in the DNA that codes for a protein. This connected to that, I guess the biological backstory is a little more subtle, but is, in computer sciencey terms, a stronger and more easy to handle hidden message. That's motifs. I'm going to explain where this comes from, what this is. For that, a little detour on why it's interesting, and that is the biological motivation. I talked about genes being portions that code for a protein, and then we talked about this process, the central dogma of molecular biology, that the genes are translated to a transfer RNA, which then in the ribosomes is sequenced into proteins. And that's how we make all these chemicals that fulfill biological functions. Now, if that was the only thing, every cell that has the same DNA would have to produce the same proteins all the time. And it wouldn't really be possible to have the same genetic information and yet have different cells do different things. And so there must be something else going on. And much of what's going on is called gene regulation, which is, I think the picture, okay, the video is Amoeba Sisters again with their particular style, but the content I think is at the right level and also accurate. And the drawings are a bit whimsical. It's a switch that can turn genes on and off. The way this works is going to be explained in a bit more detail in the video, but the kind of short version is the translation of a gene producing a copy of transfer RNA from the gene needs to start somewhere. And there's a special molecule, the RNA polymerase, that binds to a certain starting part of the gene and then starts the copying. And if you block that kind of electromagnetic starting position by putting something else there, another molecule, then that translation is not happening. Or if you make that starting region more attractive by helping it attract polymerase with other molecules, you can actually generate more of the protein. And that's what this gene regulation in the simplest case is about. Let's do the quick video here. And I'm muting my...
When I was a little kid, I had this art teacher. She had poetry, which I love. I got into on someday. And let it out. But that's creative expression. Our cells have expression too.
I'm skipping the poetry
gene expression.
motivation.
They can express genes. To express a gene means that a gene can be used to make something functional, often a protein. In our protein synthesis video, we talk about these steps. The gene, which is made up of DNA, can be transcribed into mRNA during transcription, and then used in translation to make a polypeptide chain. A protein is made up of one or more of those chains. But you know what? It's not doing that with every single gene. Not every gene is expressed. That's why the phrase gene regulation gets put with gene expression, because the expression has to be regulated. A cell in the eye has no need for using a gene that codes for stomach acid, even though the gene is present. It would be wasteful to express that gene and likely uncomfortable. Prokaryotic cells and eukaryotic cells both have genes to express. Some differences, though. Recall that prokaryotic cells just have the DNA in their cytoplasm. They don't have a nucleus. So transcription and translation all just happen here. Unlike in a eukaryotic cell, there's no traveling of the DNA out of a nucleus. Nope, in prokaryotes, it's all right there together and can really both be happening together at the same time as there's no nucleus involved. For prokaryotes, a lot of the control of whether a gene is going to get expressed or not tends to involve or impact transcription. Eukaryotes also have gene regulation that involves transcription, although they'll have a lot of other times for gene regulation too that we'll get into soon. So you might wonder, how does gene regulation involve or impact transcription? Transcription, again, is when an enzyme called RNA polymerase makes mRNA from a DNA template. There are regulatory proteins that can decrease or increase transcription, and that's important among both prokaryotes and eukaryotes. These regulatory proteins are often referred to as transcription factors. Some transcription factors bind to a DNA region called the promoter to help RNA polymerase start transcription, while other transcription factors can bind there to repress it. Some types of transcription factors can bind to enhancer sequences, where they increase transcription, and enhancer sequences can even be far away from the gene. Some transcription factors or other proteins can bend the DNA to bring the promoter and enhancer closer. Ultimately, transcription factors play huge roles in whether a gene is expressed, and we also want to mention environmental factors can influence transcription factors, meaning that the presence or absence of an environmental factor could impact gene expression. Prokaryotes commonly have operons, which feature a regulated cluster of genes along with a promoter and an operator, and they make great examples of gene regulation during transcription. We have an example of this in our Order of the Operon video, but here's a quick recap of just one type of operon known as a lac operon. Here's a repressor. The repressor blocks RNA polymerase from doing transcription, and it does this by binding to a sequence called the operator. Since the RNA polymerase can't work, ultimately these genes are not going to get expressed. That's because the genes are not going to be transcribed into mRNA, so it will not go to translation for a protein to be built. And that's great when the protein isn't needed. This is how the bacterium is regulating the genes in this operon. But sometimes the bacterium may need these genes expressed, and that's because these genes code for specific proteins, including an enzyme that breaks down a sugar called lactose. So when lactose is around, the bacterium wants that enzyme because it wants to break the sugar lactose down. It can eat the broken down lactose. So how does the bacterium turn a gene on? How does the gene get expressed? Well, as you can imagine, this repressor has got to be moved. It's in the way of transcription happening. And it turns out a form of lactose, technically an isomer of it, binds to the repressor, which makes it get out of the way. When the repressor is not blocking this RNA polymerase, transcription can happen. The RNA polymerase does transcription, and it makes the mRNA, and translation can follow, and it uses that mRNA to make protein, which includes an enzyme that can break down lactose, and other proteins too involved in using lactose. So these genes are being expressed when lactose is around. Expression is not happening when lactose is not around. It's a gene regulation example, controlling whether genes are expressed or not. And still on the subject of gene regulation that can impact transcription, we want to mention from our epigenetics video. Gene regulation that can impact transcription. We want to mention from our epigenetics video.
There's always a massive lag in the audio stoppings, but that's enough. I mean, as they alluded to, there's a few other things going on, and what the example they showed is basically for bacteria, for prokaryotic cells, where things are a bit easier. But the general principle is often the same. What do we make of this? A concrete example, which is from the book as well, why I brought it again, where they tell the story nicely, is the clock gene, if you want to follow the terminology there. How does it happen that living animals have a circadian rhythm, this roughly 24-hour cycle, that governs much of life? Now, of course, there's no, it's not a kind of universal constant that this is 24 hours. It's got to be the light of the sun that triggers this. But you usually wake up, if you have regular sleep cycles, you wake up before the sun gets up, roughly. And maybe even if not, then parts of your processes in your body start to get you ready for getting up way before the sun sets, before the sun rises. And so somehow there must be a clock started, and it's by and large started in the morning with the specific colors of light that you see in the sunrise. But then it kicks off a cascade of processes that have roughly this 24-hour duration, which can then be used to trigger things at the right time before the sun gets up again. And this wouldn't work if you were just instantly reacting to light, and then once the light's gone, you instantly get tired. Or, I mean, there's probably a lot more complicated things going on there. In animals, this seems to be more messy, but in plants, it seems there's a group of 3 proteins that have a fairly similar and consistent function in this regard. I'm not sure what these abbreviations stand for. I'm sure they have some long chemical name. I'm just going to use the abbreviations. There's one that if it's present, it means there's more translation. TOC1 promotes the expression of these other two, so more of them get made with this kind of think about ticking the block away, as in the video. Also, sunlight seems to trigger their expression. But then if these two are building up over time, and by the way, this protein synthesis, depending on how big the gene is not super fast. It might take minutes or hours to make some proteins. So this already adds a delay. And if these start building up, they themselves repress the expression of this other protein. So you have kind of a negative feedback cycle, so that over the day, a lot of these are made. They then repress more production of this TOC1 protein, which triggers more protein production of these two. But once the sunlight's gone, these start to slowly get less. That means this TOC1 is no longer blocked from the high concentration of these two guys and can accumulate overnight. And that kind of sets a timer for when the night is over. If enough of TOC1 is there, it starts promoting the production of these two again, which is then enforced again by the light. But also before then, they start waking you up in well, whatever that means for a plant. I don't know. That's roughly the story. Now, the biology behind this is super exciting. But what this means for our bioinformatics question, there must be, plants do a lot of things governed by this circadian cycle. For all of those genes that react to the day and night cycle, there must be some way how these proteins influence their expression. And that can only really work if ahead of the genes, a bit upstream from the genes, there's a conserved pattern that these proteins can bind to stop or promote them being expressed. So we should be able in plants to find for many genes some similar sequences just upstream of where the gene starts. And that could, I mean, it could be used in different ways. You can better understand how plants regulate day-night. Maybe you can identify new genes, and maybe you can tamper with that. That part we were not going to get in. And these kind of conserved sequences upstream of the actual gene, these are called motifs, or motif in general means, in this context, some shared common substring that's fairly preserved. As always in bioinformatics, don't expect the exact same string to be repeated in front of each of these genes. We'll have to live with certain little errors, mutations, and maybe also small changes that don't actually change the function, because there might be sort of equivalence classes of strings that work equally well. So the formalization that I want to look at here in the literature is known as the consensus pattern problem. I'll show you in a sec why that is a natural name. Ignore that name for the moment. Think of it as the motif finding problem. What we're given is a collection of strings. I call them G for genome, but really they're portions from the genome, which is the region just before the gene starts. And let's assume from the last chapter we know how to identify genes, so we can know, let's just read the 200 characters before the gene starts, and that's our G1 to GT. And we'll also give a length for this motif that we expect to find. And this again from the proteins that people know in plants are responsible for this. You can get some estimate. I think here you should think of K of being maybe 15 or a few dozen, not crazy large. Let's say or so. I mean, this is not a universal constant either. The goal is to find in each of these genes a starting position, so that if you read the next K characters from these positions, and you do this in all the different regions upstream of the genes. So let me do a small picture. Maybe the region you find here lies here, maybe here lies here. The positions are not really what we care about. What we care about is the content of these strings. If you write these strings on top of each other, so you should think of this being the first row here, this being the second row, third row, and so on, then there should be a fairly conserved pattern. And in formal terms, what we mean with this total Hamming distance, what we minimize here is the number of characters. So in each column, you determine what's the most frequent character, and you count the number of others. So here, this column, there's mostly T's. So 3 other letters give a contribution of 3 to the score, to the Hamming distance. And same for the other columns. It's a bit of it's maybe not the most natural generalization of Hamming distance, but that's the one that seems to be most natural for the biological problem. So if you have a column that's not changing at all, that's constant, then it's 0, and otherwise we just sum up all the columns. Is the problem clear? Okay, why is it called consensus pattern problem? Maybe you can already guess from this example. You can rewrite a bit the definition of this Hamming distance as the sum of distances in the normal Hamming distance sense from each of these red subsets, these substrings, to a specific string, one fixed string. And that's the consensus string that you build by taking the majority vote across columns. So here, because I took the picture from the book, there's other things in between that only come up later. The consensus string is this one down here. It just takes the majority letter or one majority letter of each column. And then the total score is the Hamming distance of every row with respect to this consensus string. And of course, you're just counting the lowercase letters first by column, now by rows. If you sum this up in the end, it's the same thing. All right, so the Hamming distance of the first row to the consensus would be 3, these 3 letters. And that G used to be counted here as a 1. That T and that T would contribute 1 to these counts. But if I'm summing up column-wise and then sum up those counts, or summing up row-wise and then summing up those counts, that's the same. So you could rephrase the problem here as saying we're looking for this consensus string. But it's equivalent. If you know the starting positions, you know which red strings we're looking at here, then the consensus string can be computed from that. And so that's the more specific formulation. You get the consensus string for free if you have those starting positions. Okay. I'll come back to the other parts later. The bad news first, the problem's hard, and even in a bad way. Sorry, that should be a T. It's NP hard even if the alphabet is small. They say even if it's a binary alphabet, maybe we would say the DNA alphabet with 4 letters is more natural, but that's not helping. And well, for the complexity theory people, even hoping for something that's a fixed parameter tractable algorithm in the number of strings is not going to work, very likely. No proof, but complexity theory assumptions aside, we shouldn't hope to find an exact solution to this problem in good time. Let's look at some simple things we could do, brute force options to solve the problem exactly, just to see how bad things get. One option is we're looking for these starting positions in all the strings. I remember my picture with the red parts. I'm looking for these SI, S1, S2, and so on. So that if I read K letters on each side, that has the minimum distance to a consensus string. And I could just try all combinations. How bad can you possibly get? Well, that bad. You have roughly n options in each string, n minus k. but you have to have all possible combinations. It's not clear a priori how to separately optimize in each of the strings. And so both n and t should be expected to be large in practice. N is the length of the region that we're looking at. That's certainly a few hundred. And t is the number of genes, probably also in the hundreds. So that's not going to fly. What else could we do with the reformulation of the problem from here? Maybe we can first find the right consensus string and then worry about the rest. And that indeed also works. So you could try brute force over all possible strings for the consensus string. That brings the search space to sigma to the k, still exponential, but at least k is, if anything, that's the smallest parameter of the problem, because that's the length of the region we're looking for. And sigma, the alphabet size, is 4 in our case. So maybe this is, among the bad options, still the best. What do we have to do if we guess a consensus string? We still have to find these starting positions. But that turns out to be a lot more manageable, because if you're given a certain consensus string and you have one of those GI, it is independent of what the other strings choose. The best match between these two strings will be somewhere, and you can try all the positions and just find the one that has the least Hamming distance. And the key is that you can do this independently for the different genes. So that's a bit better. And overall, still exponential, but at least no longer in all the parameters. That means that this one here, I think, is for unbounded alphabets, if I remember correctly. Otherwise, it sounds like a contradiction to this. This one indeed is somewhat practical if your motif is small. What people in practice may do is use a smaller k, find a conserved region, and see if you can grow that region manually. But it's not really satisfactory. Like you're likely going to miss more important motifs if you have to make k very small.